last lecture. We made it to the end of the quarter. It's very exciting for everyone, I think. Last thing we have to do is take the final. So today we're going to wrap up with microbial ecology, and then I have a little bonus set of slides for you at the end that I'm going to present to you that I didn't upload to iLearn, but I will show you what they're all about afterwards. Okay, so we're continuing with our discussions on microbial communities and how microbes contribute to the ecology of the planet. And so we're going to be talking about marine microbial communities first today. And in sort of a catch-all term, plankton represents the microbial communities, or a big portion of the microbial communities in a marine environment or an ocean environment. Okay? And so what does that mean? So it just means that these are passively floating organisms that are microscopic, okay? So they're anything that is below 200 micrometers in size, and then they're further categorized based on their size, okay? So, microplankton is from 20 to 200 microns, and then smaller than that are nanoplankton, which is 2 to 20 microns, and then you have picoplankton, which is 0.2 to 2 microns, and then femtoplankton, which is where all our viruses fall, which is going to be important in just a minute, is from 0.01 to 0.02 microns. So the very, very, very tiny microbes that are in the ocean, okay? And these have also been called marine snow, okay? So they're not all just floating around by themselves. They are in aggregates, and they can even be attached to other small particles that are floating in the ocean. And so you can see this collectively, and it looks like marine snow, okay? So it's like these sort of white-looking particles with a bunch of microbes attached to them floating in the ocean. And plankton is very, very important for the nutrient cycling in the ocean. So it is the number one nutrient cycling agent in the ocean are these microbes, okay, that are associated with plankton. And they're also large portions of food for other animals, okay? So how do we study these planktonic communities? Because I've told you several times already that most of the microbes that are in the ocean and other parts of the planet are not culturable, okay? So you could plate them out and try to grow them, but that would only be representative of like 0.001% of the total biodiversity that is in that environment. So you can do other things, like just take global sort of DNA content and say, okay, this is how much DNA is in this sample. And from that you can extrapolate how many cells you might think are in that sample. You can do the same thing by measuring protein and equating that to biomass. So if this sample has more protein than that one, you can say there was more biomass in that sample than the other. You can also even be really fancy and do carbon fixation. So you can show how much carbon is being fixed in one sample versus another, or in one environment versus another, but that requires that you use radio-labeled carbon, and some people don't want to do that. And then a newer technique is using metagenomics. Okay, so I'm going to explain to you what metagenomics are, and it enables you to analyze the total microbial community at the DNA level of any sample that you have acquired. Okay, and so this alleviates the need for culturing, because it's culture independent, and you just take, say for example, your liter of seawater, and you can extract the DNA from there, and then from there you can sequence that DNA and begin to assemble genomes of those organisms. Okay? So this is how it works. So this would be your example where you have the DNA from your microbial community. You extract it like we've talked about. You have to lyse the cells, obtain the DNA, and clean it up and make sure it's nice and pure. And then you're going to do shotgun sequencing, which we're not going to talk about the details of that. But instead of amplifying just single parts of a genome, you're sequencing all the parts of all the genomes of all the microbes in that sample, okay? So it allows you to do genome assemblies without having to culture the organisms, okay? And that is what we call a metagenome, because it represents many genomes of all of the microorganisms in that sample, okay? And why do we want to do this? It's expensive, it's not as fast as amplicon-based sequencing. Why would we want to do this? Well, it gives you a lot of information, okay? So it not only tells you who is there, because you've assembled all of these genomes of all of these organisms, and you can compare them to known genomes, and we can say, all right, well, we have these species that are enriched in this environment versus another environment. But we can also, because we can see the genes that are in there, because we have the whole genome sequences of these organisms. We can then start to predict what these organisms are doing in the environment, okay? So it's pretty easy to assess who is there, like taking a census of the microbial community. So that's gotten a lot easier. And the next frontier in science is trying to understand what are all those microbes doing there and how are they interacting with one another. And so this metagenomic technique allows you to do that. And so you might, in this process, discover a new microorganism that's, say, for example, colonizing your gut. But you also, on top of that, might discover some new metabolic pathways that these microbes in your gut might be utilizing to help you digest your food. So that's metagenomics. Okay, so now we're going to talk about plankton again. And one of the ways that we can study plankton is through metagenome sequencing. But I'm going to talk about more sort of a zoomed out level of what we know about planktonic food webs. Okay, and so there are lots of producers and consumers in these environments, in these marine environments, and they interact, all depend on one another, and it gets a little complicated, okay? And so when you are diagramming all of these interactions, it starts to look like spaghetti, and it's actually called the spaghetti diagram, okay? So it shows, who's producing what, who's consuming what, and then somebody else is consuming that one, and all of that. So it's this kind of a complicated web of interactions. And the take-home message one of the take-home messages from this is that in the ocean, the primary producers are the phytoplankton. Okay, so these are organisms that can photosynthesize, because they're phytoplankton, they're not the other plankton, they're phytoplankton, and they're microscopic and they're floating around in the ocean. Okay, but they are producing and fixing carbon. Okay, so they are the primary producers. Phototrophic, they take energy from the sun, and then they fix carbon, and then they turn it into biomass and carbon for other organisms to consume. Okay, so that's the phytoplankton. Okay. All right, so this is an example of a spaghetti diagram of a marine environment. Okay, again, the take-home messages up at the top, the primary producers are the bacterial and the algal phototrophs. So those are the ones that are harnessing light from the sun and converting it to biomass. And then you can see from there you have the heterotrophic bacteria that are consuming those organisms, those phototrophic organisms. And then on top of that, on top of the food chain, you have protists. Then you have other sort of zooplankton, which are like small animals that are part of the plankton. And then you have invertebrates and vertebrates. Okay? And so that is a food chain or a food web representative of what's going on in the ocean. But if you bump over to where all the purple is, okay, so those are the consumers, all right? The primary consumers in the marine environment are viruses. They predate on every level of that food chain over here, okay? And so they are responsible for most of the nutrient cycling, most of the nutrient cycling in the ocean, because viruses can attack bacteria, they can attack protists, they can attack zooplankton, invertebrates, and vertebrates, okay? So that is something I really want you to know about these spaghetti diagrams or food webs from marine environments. It's the viruses that are actually driving all of that nutrient cycling. And think about what we've learned about viruses so far. So when we talk about lytic phage, which are viruses, they get in there and they blow up a cell, right? So what's a better way of introducing nutrients back into the environment than just lysing your cell, lysing your host? Okay, so now we're going to leave the ocean and we're going to go to animal guts. Okay, so that was kind of a hard segue, but that's what we're doing. Okay, so we're going to talk about digestive communities. So we've talked about our own gut a lot and how there are microbes in there that are involved in helping us digest our food. But I wanted to use this example of the cow rumen. Okay, so cows have a very intricate digestive system. They have 4 stomachs, which the food has to pass through, okay? And the rumen is one of those. And the rumen is very critical because in that environment, there are microorganisms that help the cattle digest cellulose that is in their food. So we know that they eat hay or grain or grass, and there's a lot of cellulose material in there, and that's very hard to digest. So the cow cannot do it on its own. It does not have the enzymes necessary to digest cellulose. So it relies on its endosymbionts, which are these bacteria, those cellulolytic bacteria that are in these intricate communities that will digest the cellulose that is coming into that cow and help it convert it to energy. Okay? Yes. Sorry, I have to look at my notes here just to make sure. Oh, yeah. Okay, so look at this cow right here. So it has this hole in the side of it, right? So what the heck is that? That is crazy, but that is what's called a fistulated cow. And so this is how scientists and dairy farmers can study the rumen of cattle, okay? So the rumen is something that receives a lot of attention because rumen digestion is directly correlated to cattle productivity, okay? So dairy production, meat production, you really want to have a healthy rumen environment for that cow to be able to be as robust as it can be, and also help control several of its biological behaviors, okay? So cows produce a lot of methane, and we'll talk about that in the next slide, and that comes out the back end, right? So cow farts, that's what we're talking about here. So, and that's because of the bacteria in the rumen. So if it's enriched in methanogens that can produce a lot of methane, that can be a problem. Methane is a greenhouse gas, right? And so there are many researchers working on designing diets for cows, so when you feed them, it doesn't encourage the growth of a lot of methanogens, okay? There's also, you don't want to encourage a lot of growth of E. coli and things in there, and so we'll talk about that in the next slide, because that can end up in the food processing pipeline when they go to dismantle this cow and turn it into ground beef or something. You don't want to have a lot of E. coli carrying over from the cow intestines and guts into your environment. Okay, so this fistulated cow is a way that you can have easy access to the rumen. Okay, so they cut a hole, surgical hole inside of the cow, and then they replace it with this plastic kind of screw cap thing. Okay, so it's kind of like piercing your ear, right? And it heals, but then it leaves a hole because you put that plastic thing in there. And so you can remove the cap and stick your arm in there and take samples out when you need them. And so you can do experiments where you feed cows certain diets, and then you look to see what happens to the microbial community in the rumen, and what happens if you switch from hay to grain, or if you're enriching their diet in some way with some other form of food. Does that change the microbial community in such a way that is desirable or undesirable? And I've seen one of these before. At picnic day at UC Davis, they have a fistulated cow. They like to show everyone that cow. Okay, so this whole rumen community that we have this example here is an example of centrophy. Okay, so it's metabolic cooperation among the microbes. That's what centrophy is. And it's basically all of these microbes that might be producing some waste product. Okay, there's another microbe in there that utilizes that waste product as something beneficial. So it's like handing off your trash, but someone else's trash is someone else's treasure. Okay, so it's all of this metabolic cooperation that's happening. And if you look here at the methanogens that are circled in red, they are using CO2 and hydrogen that are produced from other byproducts of other microbes and converting that to methane, okay? And so you can construct cattle diet to sort of suppress the number of methanogens and encourage growth of these other ones that might be involved in cellulose degradation, for example, and so to really enhance the robustness of that cow, but reducing the amount of methane that they might be producing, okay? Another reason why we study the rumen microbial metabolism is because there's a concern that if you switch from hay to grain, that changes the nutrient availability in that rumen, and that encourages growth of rapid fermenters like E. coli, okay? And so that's not good, because if some of those E. coli happen to acquire some virulence factors, or that cow gets contaminated with virulent E. coli, and you're allowing for it to proliferate in the stomach and the rumen of this cow, it could end up in the meat processing portion of that cow's life cycle. Okay, so other animal microbial communities I thought you might be interested in are corals. Okay, so corals are animals, okay? It's a symbiotic relationship between, it's a mutualism, okay, between an algal symbiont and a zooxanthellae, okay? So these an algal symbiont, most commonly dinoflagellates, live inside these coral, okay? And the algal symbionts are providing the photosynthetic, and the coral is providing the shelter for that particular algal symbiont, and that is an example of mutualism, okay, where they both are benefiting from that situation. Now, coral bleaching has been observed, and what's happening is that the algal symbionts are dying off in the coral, and so they become sort of white-looking because they lose their green symbiont, and that's thought to be a consequence of global warming because you're raising the temperatures of those waters, and that is not hospitable for those algal symbionts. Okay, so now we're going to move to soil microbiology. And so this is our terrestrial environment as compared to our marine environments we were talking about earlier. And it's argued that this is the most complex microbial ecosystem on the planet is the soil, okay? And so maintaining soil health is a priority for farmers and ecologists alike. So you want to make sure that we're not ruining soil with chemical amendments or anything, because you might be trying to kill a pathogen, but you're killing off all the good microbes at the same time, okay? So this is just a cross-section of what the soil might look like. Okay, so you have at the top, you have your oxygenated, aerated zone, okay, so that's near the surface. And then you have the water table, and below that it gets anaerobic because there's not a lot of oxygen down there. And I just wanted to point out that even in the bedrock, which can be 3 kilometers below the surface of the earth, there have been microbes that have been found in there. And those are the endoliths that can actually utilize rock as a source of energy. Okay, and so we're going back up to the surface now. So we're talking about the soil, the topsoil. And if you look at any given particle of soil, it actually has a very complex biodiversity associated with it. There are millions of microbes in a soil particle. You can have bacteria, you can have fungi, you can have other organisms like nematodes in there. And it's really the microbes that are holding that soil, helping to hold that soil together, and that's an important component of soil health, is how well the soil is associated with itself. Okay, so the soil food web. Well, who are our major producers here? Okay, so it's a terrestrial environment. So those are the plants, right? So those are our photosynthesizing organisms that live in terrestrial environments, and those are our trees and any other plant that can photosynthesize. And we also have nitrogen-fixing bacteria that you learned about last time that are also major producers that are associated with plants in a terrestrial environment. Okay? But what about the decomposers? Who are they? So if we didn't have decomposers, we would just have piles and piles and piles of wood around. Okay? So that doesn't happen. So we have fungi primarily, and also some bacteria that have this remarkable property of being able to digest cellulose, but also lignin. Okay? So lignin is a very complex molecule. It's very hard to break down. Plants make it on purpose, right? So to reinforce their cell walls, and so it's very, very robust and hardy. As a consequence, it's hard to break down. Okay, so there are organisms that have evolved to do this, and so we have fungi and a few bacteria that can do this. And I just want to point out that Streptomyces species, so we've learned about Streptomyces because they produce streptomycin, right? They're a soil microbe. That's an antibiotic that we learned about. But it's also thought that the Streptomyces species give soil its characteristic smell. So like if you pick up a handful of dirt and you smell it, it has a smell, right? And those are all the volatiles that are coming off of these Streptomyces species. And you can even smell that if you grow them in culture. With it. It smells like dirt. Which is better than some microbes smell, trust me. We have a lot of stinky bacteria in our lab. Okay, so again, back to the decomposition of lignin. Okay, so we're going to talk about this in the context of fungi, because they're the primary organisms that can do this. and they produce enzymes that degrade lignin. Okay, and these are called white rot fungi. And why are they called white rot fungi? Because what they leave behind, so they degrade all the lignin, which is like the brown component of wood, and they leave behind the white cellulose. And so it's just like these pure cellulose microfibrils that you can see. And I'm sure if you've gone on a hike in the woods, you've seen these like white pieces of wood around, and you might wonder, what are those? Well, those are the result of the activity of white rot fungi on dead wood. Okay, so they're saprophytes. So you can see an example in the right here, this white rot fungus, usually colonizing a log that has fallen down from a tree or a fallen tree that is in the woods, and then after a lot of time has passed, it has degraded all the lignin, it will look like that cellulose microfibrils down in the bottom portion of that left-hand image, okay? I also want to point out there are brown rot fungi, so they target the cellulose, and they leave behind the lignin, okay? So that would be our example of the brown stuff in the top on the left-hand panel. Okay, so now also more about the soil, okay? So we just learned that soil is the most complex environment on Earth, but where the real action is, and the most biodiversity is in the rhizosphere, okay? What's a rhizosphere? So a rhizosphere is area of the soil right around plant roots, okay? So the rhizoplane are the roots, and then the rhizosphere is that small area surrounding the roots that's in the soil, okay? And why is it such a bastion of activity, right? It's because the plants are exuding root exudates that are nutrient rich, and those bacteria want those, okay? So the chemotax towards those. You learned about that last time when Paul was talking to you about nitrogen-fixing bacteria and how they're attracted to plant roots. Okay, so there are many other organisms that are also doing the same thing. So it's a highly competitive environment because everybody wants those root exudates. And so they colonize these roots, and sometimes it's a beneficial association. Okay, so nitrogen fixing, but there are also plant growth promoting bacteria for which we don't really understand the mechanism of how they're promoting plant growth, but when they're there, the plants grow better, and when they're not there, the plants grow worse, okay? So those are the bacterial beneficials in the rhizosphere, okay? But we do know a lot about the fungal associations in the rhizosphere and how they benefit the plant, okay? So we're gonna talk about mycorrhizae. So myco for fungi and rhizae for roots, okay? These are fungi that specifically associate with plant roots. And it's a very intimate association, and sometimes it's very species specific, okay? So we have two types. There's ectomycorrhizae and endomycorrhizae. Sorry. Okay, and the ectomycorrhizae do just what it sounds like. They colonize the outside of the roots, okay? So they're all their mycelia is on the outside, and they don't enter in the plant root. Whereas endomycorrhizae actually invade the plant roots in an even more intimate association. My computer does not want to move forward. All right, so this is a very good example of mutualism. All right, so the plants are benefiting because these mycorrhizae associate with their roots and actually increase the surface area of the root system. Okay, so they look like hairy sort of feeder roots, but they're actually fungi, they're these mycorrhizae, and they enable that plant to take up more water and more minerals from that soil than it would be alone just relying on its own roots. Okay, so that's how the plant is benefiting. The fungi are benefiting because photosynthate is coming from the plants into the fungi. Okay, so they are absorbing some of the plant nutrients. Okay, and so that is how both of these organisms are benefiting. And as you'll see, or in this slide, the previous slide, so the one on the left is a tree with its mycorrhizal associate, and the one on the left is the one without it. And so you can see that these trees actually rely on this. Okay, and so a lot of mycorrhizae are very essential to forest health. Okay, so if you didn't have mycorrhizal association, your trees would be less healthy. Okay, so we're first going to talk about ectomycorrhizae, and these are the ones that colonize the surface. Okay, so they colonize the plant rootlets. okay? And so you can see here in the brown, this fungal mantle. So the hyphae become very organized and associate on the outside of the plant roots, okay, into what's called a fungal mantle. And then they also have these extrusions of mycelia that look very similar to the root hairs, right? So you can imagine that if all of these roots have all of these extra appendages that are actually really good at taking up water and other nutrients, okay? Now, you can see that in the right panel here, an actual real image of mycorrhizal associated with a plant rootlet over there. You can see all that white hairy stuff is the mycorrhizal. Okay, and basically allows the plant to extend its root network, okay? And it actually has very important above ground component to it, and these are the mushrooms that you see in the forest, okay? So if you've ever gone on a forest hike and you see mushrooms coming up around a tree, it's likely a mycorrhizal association. So those are the fruiting bodies. So they send those up when they want to disperse themselves. They have spores in them, and then they release and then go and can colonize other tree roots elsewhere, okay? And a lot of these are delicacies, okay? So like truffles, for example, are an example of a mycorrhizae that is something that we actually eat, the fruiting body. And it's the reason why we can't grow truffles and other delicacies like chanterelles and things like that and morels in a mushroom farm, because you have to have this complicated association, and it has to happen in the forest. If you cannot replicate that in a farm situation. Okay, so all of those delicacy type mushrooms are actually foraged, and so there are people who go out and forage these from the forest and then sell them for food. Okay, and I will caution that if you ever wanted to do this, you should take an expert with you. There's a lot of poisonous mushrooms out there, a lot of false morels and things that could look like something that you want to eat, but they could actually kill you. So be very careful in doing that. Okay. Endomycorrhizae. So these are the ones that actually penetrate the cell, the plant cells, okay? And so this is not an easy feat without the plant trying to kill the organism because they think it's a pathogen, right? And so this association is allowed. And so you have hyphae, and they're extending out into the environment. But what they do is they form these arbuscules inside a root cell. Okay, and these are called vesicular arbuscular mycorrhizae because they form these vesicular arbuscles. Okay, and you can see that if you take a cross section of a root cell. And this inside here that actually kind of looks like a little tree is the vesicular arbuscule that's inside that root cell. Okay, and these are called VAMs for short. And there are a lot of VAMs out there that are important and very well studied for promoting tree health. And they do not form any of the above ground mushrooms, okay? So you don't really see any fruiting bodies associated with those on the top of the soil. Okay, so I put this in here because I have a very good friend and colleague who actually works in this environment here, and it's wastewater treatment, okay? And so wastewater treatment is a way that we have to clean up our water, okay? And there's a microbial component to it, okay? So the sewage gets delivered to the wastewater treatment plant, and at first it has to remove lots of solid debris, which you can imagine what that is from sewer treatment, and then you have to then fine screen, filter it, make sure you get all those insoluble particles out, and then the secondary treatment is actually microbial, okay? So you put some microbes in there that will degrade that organic content that you don't want in that water. And then you have a tertiary treatment where you chlorinate it and kill the microbes, and then you can release it back out into the environment. Okay? So this is just a slideshow of that. Okay, so the preliminary treatment, the filtering of the large debris. I actually been on a field trip to one of these wastewater treatment plants when I was like in 4th grade. But it still sticks to me today. Like, it was a really fun field trip, even though we're just like looking at poop floating around in a big tank. But it was cool. Like, at the end, if the water's clean. Okay, so then you have your primary treatment. The secondary treatment is where the microbial activity occurs, and then you have tertiary treatment. And then for now, none of this is used for drinking water. Okay, it's released back out into the environment when it's considered clean enough to do so. Okay. Now this actually happens in nature. So a a lot of wetlands that are naturally present, like the Florida Everglades, for example, are actually doing a lot of this filtering, okay? So a lot of this microbial activity and filtering of debris and things that is just naturally found in the environment occurs in places like the Florida Everglades. And there are some agricultural operations on large farms that are actually trying to mimic what goes on in these wetlands to try and clean up some of their agricultural runoff, okay? So agricultural runoff is a problem, not necessarily because of the microbes, unless it's like a dairy farm or something like that, but because of all of the nitrogen, okay? So there's a lot of nitrogen that is a byproduct of the fertilization process, and if that gets into the environment, you can have what's called eutrophication of wetlands in other places, or you have like algal blooms and things like that because you've poured all of this fertilizer into this environment that's not normally exposed to it. So you have to be careful about doing that. Okay, so this is where my colleague and his group in France is actually working on biobatteries. Okay, so this is utilizing microbes to create electricity. And so you can have microbes that act that colonize the anode or the cathode of a bacteria, and I am not an electrical engineer, so I'm not going to attempt to explain that, all of those processes that create energy, but just know that microbes can actually do this too. And what he's doing, so here, the bacteria can actually be the acceptors and donors of electrons, okay, and that will create an electric current if you let them colonize the anode and the cathode of your biobattery. And what he is doing is actually taking microbes from wastewater treatment plants to see if he can utilize some of what would be considered garbage and utilize that to create electricity. And what he's seen is that different wastewater treatment plants have different communities, which makes sense, right? And that some of them have better properties of creating electricity than others. Okay, so now I'm going to talk about the Human Microbiome Project, and then I have left myself 10 minutes at the end to talk about some plant microbiome work that we're doing in my lab. And this is just a project that has received a lot of funding from many places, including the NIH, which is the National Institute of Health, which is a federal agency. And the purpose was to take a census of all the microbes using all these next generation sequencing technologies that I've told you about to see what is on our bodies and is it different from your feet to your hands, from the outside to the inside. Are there any sterile parts of the body, for example, or things that we originally thought were sterile? Are they really sterile? That type of thing, okay? So we've learned that we have many more microbes on our body than we do human cells. And so it makes sense that we should try and study these, right? So there are many more microbial genes on you and in you than actual human genes. And so we know that they're creating lots of byproducts that we might be using, or they are doing things like digesting our food or educating our immune system that we weren't necessarily aware of before. And maybe some of these diseases, which have unknown etiology, might actually have some microbial component to them. So things that we thought might be neurological, but we can't figure out what they are. Maybe there is something that is actually, we're missing a portion of our microbiome, and that's contributing to that pathology. Okay. I guess that's all I wanted to say about that. There are some quarters where I have 27 lectures and some where I have 26. This is one where there's 26, I guess because there's 2 holidays. So anyway, the 27th lecture, which you're not going to get, has a lot of the human microbiome part. But I just wanted to make you aware that this is happening, this Human Microbiome Project, and from that we're understanding more and more about what commensal organisms we might have associated with us, and that how our microbiome changes from when we're babies into adults. And there's actually some work being done on babies born via cesarean versus vaginal birth, and how their microbiome in their gut actually changes, or is different until they're about age 2, okay? So you can predict a baby that has been born via cesarean versus vaginal birth, and think that passage through the vaginal birth canal actually deposits a lot of bacteria onto babies when they're being born. Is that important, or does it not matter? We've yet to reach a conclusion on that. Okay, so I'm going to shift over This is not